we talked about, we already had started, those of you who missed uh, influenza. So you may want to go and revisit the lecture. We're talking about some of the factors that influence the outcome of influenza. We talked about viral factors. And again, some of the viruses basically come with those receptors and they bind. And we had this conversation yesterday. Two most important receptors are present in viruses. We talked about H, heme agglutinin. We talked neuraminidase. These were the two important ones. And since they changed their sequence, kind of biochemistry, we give them a different name. So you can see all the way from H591 to and so forth. There are some homologies, some differences, but that's what happens when viruses attach. So that's one thing to understand. The other important thing that we were discussing yesterday was host factor. I said it depends upon the age, it depends upon your immune condition, it also depends upon whether you are carrying a baby. So that again is important in terms of vertical transmission. Now, as we discussed yesterday, and you can see from here, we have this neuraminidase that is required to break free from the cell that it was attached to. It kind of cleaves sialic acid and then moves away from there. And that was basically the basis of the current antiviral drug that we have. Okay, we talked about Tamiflu. Okay. Now you can also see that there are a lot of proteins being made within the nucleus. And then again, there are a lot of factors over there that we will be talking about as we move along. But also keep in mind that if you have other chronic conditions, that may also increase the susceptibility, like asthma and chronic diseases and some other chronic illnesses that we have. So everything complicates the pictures, so to speak, in terms of what's going to happen to the fate of the virus. Okay. Now, let's go to the fundamental concept of differences between A and B, right? And what actually do we mean by that, and what is happening in terms of pathophysiology? So you can see from here, from this picture, of course, so this is a virus in the center, right? And then if you're living in a system, an ecosystem, where you share the air space, and there's birds over there, they get infected with avian viruses, and there are other animals like swine, which get affected by swine viruses, and then you are there as well. Now, what you need to understand is that though we have very specific viruses that may or may not have tropism for human beings, you can see from here. So I want you to look at this picture, the words that we talked about bird flu. I want you to understand what bird flu is. We talked about swine flu. You have to understand what swine flu is. Then we talk of human flu. You have to understand what is the differences between these three different things. Okay? Now, if you know the story of the swine flu, nobody can stop actually birds from hovering around in the farms and passing on their viruses to animals. Okay? So this animal, swine, for example, in this case, can have his own virus to get infected, but it can also have another virus. The two viruses can proliferate, right, and complement each other. Now, those people who are working in farms, okay, they come in contact with these animals, so they can also share. They can pass their viruses to this animal, and this animal can pass the virus over here. So there, you can see from here, the most important thing from this cycle you can understand is that birds can also pass their viruses to human beings, and swine can also pass their viruses to human beings. human beings can pass their viruses to the swine, but they cannot possibly, as of now, pass their viruses to the birds. Okay, so what will happen is all these viruses will go and infect the cell. Okay, and you remember when the viruses go within the cell, they break open. So they break open. For example, if they have messenger RNA and they have all these protein, so there could be many things happening within the cell. We call this mixing and matching. So if you have a mixing and matching, for example, let me see if I can enlarge that. So if you go over here on this picture, let's see. So you can see that there are, again, receptors, and there are optimum temperatures that these viruses have. Okay. Now, some of these viruses may share some of the receptors because the way you mix and match the genes within the cells. Okay. So what will happen is that if a particular virus, like a swine virus, has attachment to ABC, for example, and the bird virus has an attachment for DEF, and when you put them together in the cell, they may kind of come up with another hybrid. So that's where the complications come. Okay, so you come up with another breed of viruses that go back and then look for their attachments to different sites. In this case, you can see, I don't want you to burden yourself with the name of the receptor, but understand the concept that those particular receptors which are coming from those two sources, the bird and the swine in this case, can both find their way in terms of receptors within our body. Some may go for the upper respiratory tract, as you see green color over here, as to the one that can go into the lower respiratory tract. It depends on the receptors that are coming. The other way of looking at it is that receptors can change because of the sequence of amino acids that we have. Okay, now let's see if I can go over here. So these are, for example, in the middle, this is a virus which has all those, again, heme, agglutinin, and neuraminidase, but they have these specific proteins and specific antigens in their genetic makeup that can match and mix. Okay, so you can come up with a strain unbeknown to our system and unbeknown to any part of the world, and we may have a big problem in this case. Okay, now if you were to look at the structures, for example, and you can see from here, if you pay attention, that there's a small caption over here, human, swine, bird, swine, human. So you can see when all of these are in the system, and if you were to give a color scheme, just for understanding, that the antigen receptors over here, right? For example, in this case, this is a human, right? We already know that, and it is like red, right? And then again, it is going to affect the other viruses that are specific to those particular vectors or particular subjects. In this case, you can see the closest homology that we have in terms of viruses is swine. That's why, remember, we talked about that in pathophysiology as well. They wanted to use valves, heart valves of pigs in human beings because of the cross-matching of genes that we may have. Okay, but what will happen is that once you cross-match, for example, if this cross-matches with this first, you can see it's going to come up with a hybrid, and we'll have some of the genes coming from this virus, some of the genes coming from this virus, and you can see from here in the middle. And then if it's complicated with another virus, you will see that the patterns will change. So by the end of the day, we may get a hodgepodge of all these genetic elements of viruses being picked up from here and there. Okay, and that is a struggle for virologists to figure out as to what exactly the virus genetic makeup is. And you can see from here, that's what we try to do, and there are a lot of papers published in that. But by the end of the day, you can see from here, if this was a human flu virus to begin with, and in the atmosphere, if it's coming and mixing and matching with swine and the bird, so you may end up with totally different virus where we may not have any clue as to what's going to be the outcome of it and how we're going to treat it and how do we need to make a vaccine and so on and so forth. Okay. Now, also keep in mind that A and B are the one that we normally consider them to be the major problems, but we look at C, for example. Remember when we talked of C, we talk of one specific glycoprotein, hemagglutinin esterase fusion, right? Because it has the function of both HA and NA. So HA and NA separately can be put together into a virus C. Now, flu virus has 8 segments. Remember, the segmental RNA. But the influenza variant, especially for this one, has a 7 RNA segment, right? Now, what statistically we know that all of us have been infected with virus C, and that's what pretty much is information is already there in the system. So we don't want to go, and we consider that as more or less like a common cold thing because it's not very infectious, so to speak. That's why we have no vaccine against influenza C virus. Now, this is a good video. If I have time, I'll play it up, but you can copy and paste on your own slides, on your own computers, and find out, kind of visualize how the virus comes and mix and matching whatever I'm trying to tell you in this lecture. So classic flu-like symptoms will remain, as we discussed earlier. This was a repeat information which I said yesterday, that the term flu should be reserved for influenza. We should not be talking about flu confusing with common cold or stomach flu. There's no such thing. But again, patients will say it. You'll see even in medical community, they may use that, but keep in mind it's a misnomer. So these are the typical symptoms of swine flu. You can see sudden fever, that's important, sudden cough, and then other symptoms are more like generic. But the most important thing, as I discussed the other day, is has to be sudden. Now, let's go back and look at some of the features which are common for influenza A and B. They are enveloped, 8 unique negative sense RNA. They have one protein, we discussed hemagglutinin, that is required for attachment. And it also considers the fusion protein. And again, it's going to evoke a response, which are protective antibody responses. Okay. And most of the drugs that we use, antiviral drugs like amantadine and amantadine, they basically inhibit the uncoating step and target the membrane protein for influenza A only. So these are the most important thing in terms of antiviral drugs that we have. Whereas the other antiviral drugs, like Tamiflu, they inhibit the NA protein of both influenza A and B. So these two are only for A, right? And these two, especially Tamiflu, are for both. The reason is because it targets the membrane where you have the uncoating, opening up the outer sheet nucleocapsid and let the RNA go into the cells. And the segment gene that I've been trying to tell you over the last couple of slides is where the genetic diversity is coming from. And as we discussed yesterday, most of it is mutation and reassortment of segments when you get infected with 2 different strains. So 2 different strains can come in and then cause problem. The other thing you need to know is that influenza A virus is the one that affects human, mammals, and birds. I give you an example for in mammals, swine, and then birds, again, it could be anything, but we call this disease zoonosis. Now, as I said, M stands for membrane 2 inhibitors, which are uncoating. Again, same thing, amantadine and amantadine, they only influence A and the resistance develop. That's another issue that we are having even for antiviral drugs. Whereas neuraminidase inhibitors like Tamiflu is going to affect both. So we can give this as treatment as well as prophylaxis. If one person has a flu virus A, so you want to give it to the rest of the close contacts or the physicians and nurses attending that as prophylaxis. The difference between these two is that Tamiflu over here, oseltamivir, is oral and zanamivir is inhaler. So that's another difference in terms of how we dispense the drugs. Okay? This is just a repetition of what I already told you. Now, this particular cartoon you can see again is going to take you to all those steps that we've been talking for that long for almost every virus. So you can see again, flu virus is going to go and bind first. So these are receptor first step binding. You can stop that. And then it's going to uncoat. This is what uncoating is. It's going to open it up and release those genetic. That's where those amantadine or amantadine are acting. Okay. Then we have negative sense RNA. It goes into the nucleus, transcribes into positive RNA, and then replicates, and all those protein synthesis takes place. And then all those messages which are there in the amino acid sequences will let the cell know as to what kind of the glycoprotein it needs to produce, okay? Heme agglutinin or neuraminidase, okay? And again, you can see from here, all these will be put up on the cell membrane. And then when the assembly of viruses take place, like over here, is you have this nuclear material, 1, 2, 3, 4, 5, 6. So it's like 8 segmental things that are there. And then the virus will bud out, okay? You will see that as almost a standard replication of viruses for different viruses in your book. And again, 1, 2, 3, 4, 5, you can see we have that into different steps. And this is basically a legend for whatever was there in terms of the figure. Now, yesterday I said antigenic drift and antigenic shift, okay? And I also told you that I gave you an example that you're driving in a car and you just sleepy, you just wave around your drift, right? So that's like a minor thing, right? And I also said endemic. I must have explained them hundreds of times, but one of the questions today for bioterrorism was saying the endemic rise and fall. Endemic sudden rise and fall. Endemic doesn't rise and fall at all. Epidemic rises. So that's a basic understanding. You have to understand the difference between A and B and what do you mean by endemic and epidemic. That's the minimum information that I would want you to gather from statistic class. I'm going to talk to Kumar as well. Okay. So minor. So this is a minor change that you will see. Mutation of HA genes, and this process takes 2 to 3 years. Remember when I gave you the timeline of viruses? We used to wonder where the virus disappeared, why does it come back, and what is going on with the virus. So in this case, this process, which is like I'm explaining to you in a second, it takes years to cause the local outbreak of both influenza A and B. So drift is taking place in both A and B, which is a minor thing, okay? And again, this is remember I said yesterday that these are the CDC charts for public information. So when you go next time, whether you want to take your time away from your iPhones and start looking at whatever they have put over there, so you can see from here, this is for public information. They are talking about those receptors expressed over there. They're talking about these antibodies that bind it. You can see this antibody binding. And sometime, if, for example, this particular virus mutates, it kind of mutates one of the HA or NA, so antibody would not be able to bind with that because antibody binding is specific. So this kind of mutation that happens within the genetic change that you can see from here, that the shape of those particular glycoproteins are changed, so antibodies would not work. So this kind of change we consider as a minor, and we call that the antigen has changed a little bit, drift. So that is a drift that can happen in both A and B. Again, public information displayed on for patients. As far as we are concerned in this class is that when we talk of shift, for example, antigenic shift, what is happening is that now this is reassortment of genomes among different strains. So that was happening within one strain. Okay, and this is happening where you have different strain, including animal and strain. So that's why this process only takes place in a virus. Okay, so if I ask you, if you have swine flu or bird flu, what virus are we talking about? We're talking about A. Okay, because that's where the mutual exchange, and that such changes are often associated with pandemics. So whole world is involved. That's why you see this WHO charts over there. That is a major change, major antigenic change. It involves multiple hosts. So there are like three important, there's a mnemonic for that. So major from multiple hosts, and then again, it is basically pandemic, is for the world over. So these are the major things that you have. Again, you will see another picture from physician's clinic where we want to educate the people. And you can see from here, in this case, what's happening is bird, swine, and again, the same story that I tell you, both of them can pass on information, genetic information to human beings, and we may get a mixture for that. Most of the time, what happens is, you can see from in this slide as well, the way the transmission takes place is that the birds are going to go and infect swine, and then swine will have both of these viruses within itself and then come up with a new virus. And then the farmers raising in pig farms, they're going to pass it on to human beings. And you can see all those changes that may happen where you kind of mix and match two different viruses. So this kind of a thing is scary, and we call that as a major antigenic shift, and that probably is the threat that we have. And we also, and there's another one. I mean, there are tons of them. I mean, I just picked up some of them. So keep in mind that many of them even are shown, I know that, I shouldn't say I know, that when did you guys do your high school, but in some of the high school biology as well, they show them these pictures to tell them what is flu and what is going on. So you can see very simple, but a very good one, and that's very self-explanatory. Let me see if I can expand that. So you can see from here that there are different methods of transfer for that. So number one method is that a bird strain of influenza A can jump directly from a duck or aquatic animal to human. We call it bird A influenza. And then it goes to the human host, and it can mutate by itself, because viruses are known to mutate. And then strain may evolve to spread from person and to person and person. And so this way also a pandemic could rise. That's one of the ways the pandemic could rise. The other thing is that the other method over here is that the bird flu influenza A infects an intermediate host, which is a pig. So you can see over here, so that's the second type of method. Again, it's going to get mutated, and then this mutated virus can come to affect this human host, and then human host will spread it up to the rest of the community. So that's like a method 2. Now, thirdly, so you can see from here that the bird host and the human host, they have 2 different strains of viruses, and both of them happen to be present at the same time in pig, like intermediate host, and you come up with a new influenza strain. So if you come up with a new influenza strain, it's going to go and affect human host, and you will have a mutated influenza strain affected to these. So you can see from this method, 3 to 1, there's another mutated influenza, and there is a third one also. 3 methods of mutation, but you can guess that if you get more mutations from intermediate hosts coming from 2 different viruses, the chances is that it's going to be more of a problem. Okay, so it's very educative in terms of visualization of the things. Okay, well, if you the same concept, if you want to read it from your book, you can see from here that you have a specific virus over here with all those sequences of genetic material, right? This is type 3 transmission. And then you have a human, which also has a different type of sequence of genetic material, like 8-segmental RNA. And you kind of put them together into intermediate host in a pig. And then you can see from there, it's going to be mixing and matching between these 2 colors, and you will come up with a recombination of all these things and come up with another virus, which is totally weird and is going to then cause a problem. So this is where we get the shift, and all those numbers which are attached over here, like H3, N2, H5, N1, there are different kinds of sequences that we have where there is a reassortment of mixing and matching taking place. Okay, now as far as the clinical picture is concerned, again, the big arrow, major contribution to pathogenesis and immune response and less frequent outcomes. So you can see from here, you get aerosol, it's going to go, same story, going to go to respiratory tract. And then again, at that level, there will be immune response. You can see from here, which means interferon induction, which may mean production of antibodies, the T cell responses. So these will happen over here when you have that virus in your respiratory system. Then again, it can go from there and then cause what we call influenza syndrome. So influenza syndrome will be when a lot of different body parts are involved, and you like get pneumonia, you may have CNS problem, muscle problem, so on and so forth. So classically, influenza would mean that respiratory tract and flu-like symptoms, but eventually, when the time progresses, you may have a generalized immune response that you normally see. Okay? Now, once your system sees over here, right, interferon is a genetic response, so antibody and T cell responses are responsible for the future protection from that particular virus that your system is fighting for. Okay. This is basically a legend of whatever I just told you in the previous slide. And some of that is, again, immune response to viruses that we already have discussed in immunology part. cell-mediated immune response, immunopathogenesis that we've been talking about for the last couple of days. And a little bit recap of your immunology part. So you have a cellular response, virus goes into the body, and this virus is presented in context of MSC class 1, and then the T cell will come, T memory cells, and T cell will help plasma cell to make antibody, typically response that we discussed as viral proteins be presented through MSC class 1 pathway. If you look at the virus growth, that virus grows into our body. So you can see that we talk of virus growth in primary, secondary, and tertiary. So you can see that kind of goes and wanes over time. But at the maximum, it's going to be like 10-day cycle. So most of it, like primary growth ends at 10, secondary growth ends up like 7 days, and the tertiary growth ends like 5 days. Whereas the response, so what do you think, what kind of response should you get in terms of a cell-mediated response to fight viruses? Sorry? Delayed isn't hypersensitivity. Viruses are not hypersensitivity responses. What kind of cell-mediated response that you think you're going to get to fight viruses? Yes, the answer is there. You just have to look. It's just cytotoxic. You're right. Cytotoxic, CD8. Because remember, when something is presented in MSC class 1, CD8 T cells. So we need CD8 response. So you can see from here that response sets in. A primary response takes a little bit of longer time, and then a secondary response. So we have to have CD8 cells in terms of fighting off the responses for viral infection as compared to bacterial, where we may have CD4. Now, this is a time course of influenza A virus. Again, you can see in terms of days. And what you would see is that there is an incubation period. And then again, the very first thing that you notice over here in terms of how we move to the right is virus is present in respiratory tract. And then you have interferon induction. Whatever you saw on the graph, if you were to put it in the clinical picture, you will see most of the time you have a primary infection over here within 6 to 7 days, and you may get secondary bacterial infection. So you may get a secondary bacterial infection, which may come later, okay? And then depending upon your immune response, you will see that what we normally do is, in terms of laboratory detection for these viruses, we can detect viruses in the first 7 days, right? And these viruses are there. So if a person comes with the flu and you find out, I ask the person, when did this symptom start? 8 days. So there's no point for you to look for viruses because that person may not be passing viruses in the secretion. No, you're going to look for virus-specific antibody in terms of pathogenesis. If you look at, for example, the clinical picture, you will see that it's a symptomatic initially, and then you may have a bacterial infection, and then the person may go to convalescent stage over time. Okay? I think it's also about time. We are not introducing, I mean, we are kind of, but there were some questions, clinical picture, in the study guide that was posted to you. And those of you who did go through the question, they must have realized that I'm not going to give you percentage, but quite a lot of questions were there from the study guides posted to you. They were from there. I just copied it from there. So you can even go back and look it up. The thing is that some of them are like, clinical questions. For example, you say a patient walks to you, he has a flu, and the symptoms started yesterday, and he wants to ask you, what type of test are you going to go for that? Are you going to look for virus in the secretion, or are you going to look for antibodies? And I just gave you a clue over there, second day. Okay, so this type of the questions are usually clinical case questions that you really have to get used to it, because most of the time the NAPLEX type questions or the questions that they want you to train with are in clinical perspective, asking the same thing. Okay. All right. We did talk about epidemiology, and again, I think I've already talked about that. The most important thing is that when we look for the strain, for example, B, we're going to look for the type, geography, and type of isolation. So you can see if you go into literature, they're going to talk about, sometimes they give, if you pick up that particular infection in Singapore, they may call it Singapore strain, or you may call it China, whatever. Right? So they sometimes pick up the words over there, or sometimes they say H1N1, they give you the actual protein marker or glycoprotein. Now, this virus is a large virus, envelope variant. I already told you that there's segmented genome, and I also told you that 8 segments, C has 7 segments. That's very important for you to know. And then again, we already talked about influenza A, vertebrate species, as compared to B. We already discussed that where is this genetic reassortment coming from? So the genetic reassortment is coming because of co-infection with animal and human strains of influenza. So you saw in a pig where that pig got infected from both, from bird flu and human flu. So that was the worst part in terms of us having a very different virus strain, right? And the other thing is, if you look at the clinical picture, transmission of virus often precedes symptom. That's pretty much the rule for most of the viruses that I've taught so far. Now, spreading of virus transmission, again, droplet, talking, breathing, coughing, and then again, it is a seasonal as well. Virus is extensively spread by school children. That's why it is a public health hazard. And I mean, I told you, I don't know how many times that if you call the attendance line, the voicemail, you say if your child is sick or having a flu-like symptom, you want to keep it at home, please do so. You don't want to infect other people. So who are the people at risk? Well, we talked about the risk factors already to begin with. Those people who are seronegative, this means they never have been exposed to that particular virus, so they don't have any protective antibodies. Adults, children, high-risk group, elderly, immunocompromised, and people who may have chronic problems, cardiac problems, and so on and so forth. The other risk factors are, again, chronic diseases and some of the things that we normally see: immunosuppression, sickle cell anemia, hemoglobinopathy, aspirin therapy, especially in rheumatoid arthritis, because aspirin is going to be anti-inflammatory, and you want a little bit of inflammation to mount a good immune response. If you have diabetes, renal, and metabolic disease, pregnancy, and again, senior citizens are vulnerable to this. How do you control it? Now, remember that I said that some of the drugs are approved for prophylaxis or early treatment. Sometimes you want to treat the people in the house, and we have these drugs available. We did talk about what the functions are. These two are M2 inhibitors, where the others are NA inhibitors. Vaccines are available, but they are predicted yearly because of the then the question is, we don't want to immunize you with swine flu, or bird flu at this stage, because there's no need for, there's no outbreak. But they already prepared themselves in advance if the need be. What do they need to do? Again, symptoms that we normally see associated with influenza virus: acute infection, and you may get some of the complications. We already discussed that earlier. That it depends upon only viral infection, or secondary bacterial pneumonia. And all other things that we have been discussing so far depends upon the attachment and tropism of these viruses. As far as lab test is concerned for influenza virus, again, the classical is a cell culture, monkey kidneys, I told you. And I also told you, I think yesterday I told you 4 different types of cytopathology. And guess what? I want you to know, I'm going to ask you that, right? What are the cytopathological changes that happen in viral infections? But if you were to look at the other markers, for example, hemagglutinin is going to test presence of virus, and you have some antibody production. You can also look at them. The whole idea is the serology is basically either looking for the antigen or antibody. So all those tests that are there for antigen, for example, PCR, are important as compared to ELISA, immunofluorescence, which are for antibodies. Treatment, prevention, and control, again, other than the symptomatic treatment that we have, like Tylenol, antihistaminic, the antiviral drugs or its analog are available. And all you need to know is that they are M2 inhibitors, uncoating step of influenza A, but not B and C. Right? So this is a very specific protein we call M2 protein. And then we have the inhibitors for neuraminidase, because remember, as I said yesterday as well, that the heme agglutinate of the virus, it binds to nucleic acid on the viral particle and forms clump. So this particular drug is working on that. So it kind of, and it's also available orally in this version and inhalation in this version. So it's an inhibitor for the viruses that would not allow it to break away from the cell and infect other cells. But again, if you look at the picture, you'll also see that it's not going to protect much of cytopathology because it's the terminal stage of the disease. What can you do in terms of travel, in terms of people being confined into smaller places? There's nothing you can do because you cannot stop that happening. The natural immunization is there, but again, it's not protected for a long period. These strains are available, and then again, they will do their best if you were to travel to those countries where they have endemic. So endemic means always present or more or less stable rate. Right? So you want to go over there, so you may need to get a antiviral drug prophylaxis. So what they will do is that they will pick up the strain of that year that's prevalent over there and give you a vaccine for that. The other important thing that you have to keep in mind is that I'm not going to give this assignment to you, but last year we gave this assignment to the students to find out how the vaccine is made. But those of you who are interested, they can still Google and find out that how do they make these vaccines and what's the time lag in terms of productions. But one thing I think you must have known already, that these are prepared when the viruses are grown in embryonated eggs. So they grow them in those embryos of the eggs, and then there's a very nice video available, and then kind of inject viruses into that, virus goes, and then they inactivate those viruses, and then they go purify that and make the vaccines. But there's a timeline attached to it, and it's done very diligently, and you have to take care for all these precautions. And then again, these are the recommendation from CDC that these people should be vaccinated, and those of you who work in vaccination clinic in these different pharmacies, they are pretty much aware with that the person with allergies to egg should not get the vaccine, because vaccine is made in embryonic eggs. So that's where it is coming from, and it's also a protein. Okay, and again, also some of the questions, like for example, if I ask you that why do we not give this vaccine to the people, and what is going to be the outcome of that if you do that? So most of the time, it is nothing to do with the virus per se or attenuated virus. Is more to do with the protein response for allergy. So that's what the problem is in this case, because is an allergic reaction. Right? So that, but again, we already talked about in immunology about desensitization as well. If there is really a need for us to do that, there are other ways that we can circumvent that problem. Okay, now recently they're talking about a nasal spray, and I don't know how, did anybody get nasal spray? Did anybody try that? You said yeah? You yourself, or the doctor prescribed you? Okay, okay. So at least we have one person who tried that. So, so the thing is this, that you're not talking about. Okay, that's fine. Don't worry. But what I would want next time when you do, make sure that you read what is it, because a trivalent vaccine, which has all those HA and NA gene segments, right? And they kind of adapt that to temperature, and most likely this will elicit a natural protection. So this is like the thing that we want to kind of encourage for people to have. This is recommended for people 5 to 50, and again, depending upon if you are in those CDC recommendation group. Otherwise, again, don't get panicked. So this was part of that exercise last year that we wanted the students to do. That you can see from here, annual influenza vaccine production is a timeline, and CDC keeps that timeline, and then again, it takes you from all those steps all the way from finding out, hey, where the infection is. Okay, let's go and sample the virus. Let's kind of prepare that, and you prepare, purify, assemble and deliver. You can see all these steps are there as a emergency preparedness. So you need to keep in mind, many times you'll go to the doctor and say, I want to have this flu shot. It's not available yet. So CDC has its timings when the next shot will be available. So don't expect that you get a CNN news today, epidemic broke, and then you start calling CDC. It's going to take time. But they are prepared for that. So they have dedicated staff to really work overtime and kind of, diligently to make sure that it's provided as a public safety measure. Okay, last couple of slides from, again, these are, you remember when I say new drugs. When I tell you new drugs, what I mean is that if you have to go to, this is the famous, and I'm pretty sure those of you who did go, I'll find it out when I look at your individual project, that if you open up drug discovery Nature reviews, they usually give you, that was your exercise to find out what is there in the pipeline. And I can assure you, for every microorganism that we gave it to you, there are drugs in pipeline, not necessarily available, but being talked about. So you can see over here, all the way from the first step, which is here, where we have the very first step that a virus wants to attach to the cell. We call adsorption. We have H and N and M2 protein, and the receptor containing sialic acid. So you can see the sialic acid receptor is there. So there are drugs that we're working at this step. Then we have these kind of derivatives which are going to work on endocytosis and fusion when you have to kind of draw this in, and then uncoating. And then you can see from here we have this transcription process, budding out, packaging and budding out, and some of them over here, like I just gave you, neuraminidase inhibitor, which is a Tamiflu. So you can see the steps are the same, but if you look at the drugs, for example, this was just to show you neuraminidase inhibitors, but I just gave you an example for only 2, but there are more than 2, right? For example, they want to improve the efficacy of that, delivery of that, and so on and so forth. So these are some of the things that are available, and you can see that for every step, they are working on inhibitors, all these things, because we want to stop the replication of cycle. And as I may have mentioned many a time, it takes 10 years and $1 billion for a pharmacy company to come up with a drug. So they already may have invested tons of money on there, okay? But you can see by the end of the day, still receptors are involved, and blockers are involved, and drugs are involved, and we just want to make sure that some of them are available. The last slide, I mean, you can see from here, eventually, not me, but eventually what they would want you to know is the actual structure element of the many of drugs which may act on those particular areas over here. So you can see that they talk of purifying the drug, they talk of those specific receptor attachments and so on and so forth. Again, if you look over here, for example, if I can go, so you can see that the story looks pretty much the same. There is a neuraminidase cleavage receptor, neuraminidase inhibitor, and we have hemagglutinin. And the whole idea is that we want to block that. And that's why we come up with different type of drugs. And you can see this one is acting like H3N2 receptor at the upper respiratory tract, and this one is H5 receptor. So you can see a little alteration of the carbon chain or ring over there, and substitute one of those molecules can make a major impact on that. So that's where you want to come from, in terms of drug delivery and whatever is there in terms of us looking at that. Okay, so that pretty much